Hello students, a very good morning to you. How are you? I know you all okay and doing your homework very properly. So very good students. I am proud of you. As we know, we are revising our chapters. We already completed the revision of chapter 1 and today we will start the revision of chapter 2 of class 12, that is sexual reproduction in flowering plants. Remember, this is the most important chapter in class 12. This is the base to know the sexual reproduction in plants. As we know, the plant life cycle is, you think the plant life cycle is little harder than the animal life cycle. But here, nothing seems like this. If you watch the videos carefully and listen attentively, then follow your NCERT book, then there will be so easy to understand the chapter. So let's start the chapter, that is revision of chapter 2, sexual reproduction in flowering plants. As we know, sexual reproduction is normally seen in higher plants. Like some species of pteridophyta also shows sexual reproduction, bryophyta also shows, but the main reproduction occurs in angiosperms, or which are called as flowering plants. If any plant bears flower, then we will confirm that that plant reproduces through sexually. How? Because flower contains the sex organ for the plant. That's why the flower is the most fascinating organ. Okay? Flower is the most fascinating organ of the plant as it contains the sex organs. Not only the flower contains sex organs, but also it gives us aesthetic pleasure. We produce perfumes from it which we soon on our body. We enjoy the beauty of flower. All those things are given by flowers to us. We are so lucky that we are human beings and we have a large variety of plants which bear different type of flowers. So students, how these flowers reproduce? Which parts help in the reproduction? Which parts get converted into fruit that you eat? Which parts bear nectar or fragrance? Which part protects the flower? And although how we will able to know the reproduction, the sexual reproduction occurs inside the flower. So students, let's see it through a diagram. As we know, depending on according to the presence of sex organs, the flower is divided into two types. One is unisexual. I will not discuss more about this as I have discussed in chapter 1. One is unisexual and another is bisexual. So here let's see the flower, that bisexual flower and the organs it contains. You can see here the bisexual flower. This is a bisexual flower. You can see the diagram. This is the diagram of a hibiscus or China rose. This is diagram of a LS, longitudinal section of hibiscus. Hibiscus, or we can say as China rose. So what are its parts? This part is called as calyx, means the combination of sepals. Sepals combine to form calyx. They are green leaf-like structure. Okay. Next, above the calyx, there are petals and form corolla. Petals form corolla. Next, move to the female part. This is called stigma. This part is called stigma. This part is called style and this part is called ovary. So stigma, style and ovary, they combiningly form the female sex organ that is called carpel or pistil or gynoecium. Called carpel or pistil or gynoecium. Clear? Stigma, style and ovary represent the female part and the female part is called carpel, pistil or gynoecium, whereas this part is called as anther and this part is called as filament. So anther and filament together form the stamen. This is represent the male part in flower, or we can say as androecium. So this is male part and this is female part. This is male part and this is female part. So the diagram we made or draw is a bisexual flower. Here are sepals. Sepals protect the flower during its bud stage. Means when the flower is not bloom, means not open, then at that stage the sepals, which are green color structure, leaf-like structure, protect the flower. Next, the petals, which contain the fragrance and nectar, due to which pollination occurs. And they are variant colors like red, yellow, pink, white, etc. etc. Different color petals we can see. Next, here is the male part, inside which pollen grain is formed. Pollen grain is formed which carries the male gamete, and inside the pistil or carpel or gynoecium, egg cell is formed. So the main part of male and female is pollen grain and egg cell, which participate in the reproduction. As we know, when we are studying about sexual reproduction, then the sexual reproduction is completed through 3 steps. Sexual reproduction is completed through 3 steps. That is pre-fertilization, then your fertilization and post-fertilization event. We know it. First pre-fertilization, then your fertilization and post-fertilization. We will not discuss more about this as we have already discussed in chapter 1. So first we will study here the pre-fertilization event, and in pre-fertilization event the first event is gamete formation. So let's study about gamete formation. As we know, in sexual reproduction 2 different types of gametes are formed and participate in reproduction. So the 2 type of gametes formed in the plant is, one is male gamete and another is female gamete. So the male gamete formation occurs by the process of microsporogenesis. And female gamete formed through megasporogenesis. So we will study about microsporogenesis, then megasporogenesis. So students, let's study that pre-fertilization events. First we will see that which part of the flower contains the male gamete. As we know, the anther contains, if we see the anther, then the diagram of anther is like, this is anther. This is an anther. So this is called filament and this is called anther. Inside anther, pollen grain is formed. How this is formed, we will see. So in angiosperms, the anther is bilobed and diathecous. In angiosperms, angiosperms are the highest plant species in the world. Angiosperms, anther contain the male gamete and it is bilobed and diathecous. Bilobed and diathecous means, what do this mean? This means if we cut the transverse section of anther, then we can see the diagram like this is the structure of a anther. If we cut from a transverse section, then we can see like this. This is the anther, we can see. So The anther has, as I have told you, bilobed and diathecous. Here is the lobe, one lobe, and here is another lobe. So they are bilobed, and each lobe contains two theca. Theca means shaft, inside which pollen grain will be formed. So this is lobe, this is one lobe, and this is one lobe. So 1 plus 1, 2 lobe, and each lobe has two theca. Here is 2 and here is 2. So if we see the diagram of a anther, then we can see that the anther has bilobed and diathecous structure, and the theca contains the pollen grain and the pollen grain contains the male gamete, which further take part in the sexual reproduction. So students, let's see what are the different parts of the theca and what they are called. You can see the diagram. Next is, let's see the different parts of the theca. We are studying about pre-fertilization event. As we know, the process is called microsporogenesis. Microsporogenesis. So, before we study about microsporogenesis, as I have showed you the diagram is like this. You can see this is anther. Clear? So each of the theca is called a microsporangium. Microsporangia is the plural. They combiningly called microsporangia. Microsporangia. Whereas a single theca is called microsporangium. Microsporangium. So let's see what are the different parts of a microsporangium through diagram. So students, if I show you the single microsporangium, then you can see its different layers. Means suppose this layer, I am representing here one of the microsporangium. We can label the different layers. First is epidermis. This part is called epidermis. Clear? Just below the epidermis, endothecium is present. This is called endothecium. Normally, below the epidermis, mesothecium would come, but here, just below the epidermis, endothecium is come. Next is called your middle layer or mesothecium. Middle layer. Then just below the middle layer, there is presence of sporogenous tissue. This is sporogenous tissue. And inside the sporogenous tissue, there is tapetum is present. Sorry, this is tapetum. And inside tapetum, sporogenous tissue is present. This is sporogenous tissue. So let's understand in detail. I have drawn a single microsporangium. This microsporangium I have represented here. All these 4 microsporangia have the same structure. The outer layer is called epidermis. The outer layer is called epidermis. Let's understand. Okay, you can see. This layer is called as epidermis. Just below the epidermis, here is the endothecium. This layer is called as endothecium. Next, below the endothecium, there is presence of middle layer or mesothecium. You can clearly see epidermis is the outer layer. Then just below the epidermis, the endothecium is present. Just below the endothecium, the middle layer is present, and below the middle layer there is presence of tapetum. There is presence of, this is tapetum, and this part is called your sporogenous tissue. Just below the tapetum, here is the sporogenous tissue. So let's see what is the function of these layers. We can see the same layer in each of the microsporangium. Clear, students? So the epidermis, endothecium and middle layer gives protection to the sporogenous tissue. Why? Because the sporogenous tissue divides to form the pollen grain later. Is that clear? The sporogenous tissue divides later to give pollen grain. We will study in detail through diagram. You just simply try to understand here that what is a microsporangium contains. And a angiospermic anther has bilobe and ditheca. Each theca is called as microsporangium, and a single microsporangium contains epidermis, endothecium, middle layer, tapetum and sporogenous tissue. As I have told you, epidermis, endothecium and middle layer works or gives protection. They are the coverings of the microsporangium or microspore mother cell, which we will study. Then the role of tapetum. Tapetum is made up of tapetal cells. Means they are the pre-nuclear present around the sporogenous tissue and they give nutrition to the sporogenous tissue. Remember, tapetum is made up of tapetal cells. Tapetum is made up of tapetal cells and they give nutrition. To the sporogenous tissue. Give nutrition to the sporogenous tissue. As the sporogenous tissue further divides to form pollen grain. So how this process occurs? Let's see. The process of formation of microspore tetrad is called as microsporogenesis, which is the topic of today. Microsporogenesis, the process of formation of microspore tetrad is called microsporogenesis, from which it will form. Students, remember, the sporogenous tissue has the ploidy 2n. Here I have written the sporogenous tissue and it has the ploidy 2n. First, what happened? The sporogenous tissue goes through mitosis. Remember, the sporogenous tissue goes through mitosis for its differentiation. Clear? And through differentiation, it will give microspore mother cells. So here the sporogenous tissue goes through mitosis and gives 2n, which are microspore mother cell, MMC, or we can say as PMC, pollen mother cell or microspore mother cell. They will, after differentiation, means when their number is so much maximum, then each MMC, microspore mother cell or PMC, goes through meiosis to form microspore tetrad. So how this happened? You can see. Here is the sporogenous tissue whose ploidy is 2n. Means this is diploid. First it goes through mitosis and form 2n. They are called as MMC or PMC, pollen mother cell. Again they are go through meiosis. Meiosis 1. Through meiosis 1 they form half the number of chromosomes, means N and N. Again through meiosis 2 they form 4 haploid gametes. This is meiosis 2 and I have told you that meiosis 2 is equivalent with mitosis. So here equal number of chromosomes are distributed among the tetrad. So as here formed is 1, 2, 3, 4. So this is called as microspore tetrad. They are called as microspore tetrad, which contain the further they converted into pollen grain. So students, as here the sporogenous tissue divides into many microspore mother cell. So again they go through mitosis and they form large number of pollen mother cell or microspore mother cell. So the pollen grains formed inside the microsporangium is large in number. If we cut the transverse section of a anther, then we can see yellow color powder like structure. They are called as pollen grain. How they are maximum in number? As they divide first mitotically. Remember student, first mitosis will occur in sporogenous tissue. Then they give microspore mother cell or pollen mother cell, and that microspore mother cell goes through meiosis to form 4 haploid gametes, which is called microspore tetrad, or we can say as pollen grain. So this is how pollen grain is formed, and reproduction occur in plants, means how male gamete is formed. So the labeled part you can see here. You can enlarge the diagram and should practice the diagram. Each topic in this chapter have the possibility to come as 5 marks question in your board exams. So you can't ignore any topic and should attentively learn this. Can take the screenshot, and if you have any doubt then you can ask me. For today this is all about your microsporogenesis. As I have told you, formation of microspore tetrad from MMC or PMC. MMC, microspore mother cell. PMC, pollen mother cell, whatever you can call it. So students, this is all about your microsporogenesis. In the next class, we will study about pollen grain.